Good evening, everyone, and Happy New Year. Thank you very much for coming over for our first installment of Café Scientifique Harpenden for 2025. Today we have Dr. Andy Gregory, who is going to talk about long-term experiments. Andy is a soil scientist at Rothamsted and is the head of Rothamsted Long-Term Experiments, National Bioscience Research Infrastructure. In his role, he coordinates the maintenance and development of world's long-running agricultural field experiments and its associated sample archives and databases. He completed his undergrad and postgraduate degrees from University of Leicester in the 90s and doctorate from Cranfield University. His main research interests are in interaction between soil carbon and soil and physical properties. He's a member of the British Society for Soil Science and a member of the editorial boards of two soil science journals. I hope all of you enjoy his talk, and we're going to continue with the Café Scientifique talks. We have several lined up until May, and the next one is going to be on the 11th of February. So thank you, and welcome, Andy.
Okay, thank you very much, Anu, and thanks everyone for coming out tonight on a Monday evening in the winter. I'm Andy Gregory. I'll tell you a little bit this evening about the Rothamsted Long-Term Experiments and how these 19th century trials, because that's probably all they were initially, that's what they were envisaged to be, how they still continue to support 21st century science. I think it'll take about 45 minutes if I don't test people's patience, so I'll try and read the room and see how many people are still awake and tailor it. But I'm going to set myself a bit of an alarm and see how I get on. Of course, no talk about the history of Rothamsted or the long-term experiments can start without mentioning one person, and that's John Bennett Lawes. So Lawes was born at the manor at Rothamsted in 1814, but his father died when he was only 8 years old, so he actually inherited the estate aged 8 in 1822. He was educated first at Eton College, and then he went up to Oxford at Brasenose College, so that gives some indication of his standing in society and the family's standing at that particular time. Now, he only stayed at Oxford for about a year. He didn't actually graduate, which wasn't unheard of, as we say, for a person of that place in society. But actually the main reason, age 20, he actually took on responsibility for the Rothamsted estate in 1834. But he had stayed at Oxford long enough to get interested in nutrients, what nutrients were important for crop growth, indeed how crops actually acquired the nutrients in the first place. There was a lot of unknowns in the mid-19th century. It still wasn't clear precisely how plants acquired nitrogen, whether they acquired it through the air or through the root systems. So a lot of unknowns. He was very interested in phosphorus. I think phosphorus, already well known as being an important macronutrient for crop growth, and it was already well known that you could extract phosphorus from bones, a lot of phosphorus in bone material. You could actually dissolve it, extract it using acids and to form a powder that could then be applied to the land, and that was already well known at that point in time. But in the late 1830s, leading up to the 1840s, on the Rothamsted estate, we have got documentary evidence that Lawes was already setting up little trials, little pot experiments, exploring the effects of some of these inorganic fertilisers on crop yield. And as we say, the phosphorus was well known and the means of acquiring it was quite well known, but what Lawes did was to patent a method for producing it at scale. So he took out a patent in 1842. He had to defend it on numerous occasions with people trying to copy him. And he basically set up factories to produce superphosphate, the world's first commercially available inorganic phosphorus fertiliser. He had a factory in Deptford on the Thames, a second factory in Barking followed, and he manufactured and supplied fertilisers, not just phosphorus, but also nitrogen fertilisers as well. And that brings us really to 1843. And I guess he'd probably come to a period in his life where he had the scientific interests in nutrients. He had a successful fertiliser company, so he had money, and he had a large agricultural estate here at Rothamsted. So the time was ripe to formalise some of these experimental trials that he'd already begun to kind of tinker in. He established what we know today as Rothamsted Research. I think initially known as the Rothamsted Experimental Laboratory. For many decades it was the Rothamsted Experimental Station, those of you who've lived in Harpenden for a long time. Began to set up the first of the classical experiments, which I'll mention in a moment. And the other important event in 1843, as far as Rothamsted's concerned, he appointed Joseph Henry Gilbert to be his laboratory manager. So at that point, it's worth perhaps just pausing and go back to 1843 and what condition the UK was in. Well, the population of Great Britain was only about 18.5 million, plus another 8 million on the island of Ireland, which at that point was part of the United Kingdom. But life expectancy was only about 40, 42 for women. Queen Victoria's on the throne, still a very young woman. She'd been on the throne for about 6 years at this point. The Prime Minister, Sir Robert Peel, perhaps best known for founding the Metropolitan Police, and that's where the informal word bobbies comes from. And then in science and technology dominated by father and son Brunel. Father opens the first tunnel under the Thames, and the son, Isambard, who came to eclipse him in many ways. Well, the SS Great Britain was launched in 1843, and you can still see that today in Bristol, of course. And in science, James Joule experimentally finds the mechanical equivalent of heat, and the name Joule lives on as a unit of energy or heat. And in literature, Christmas Carol was published in 1843, which is a bit of a mainstay on the television every Christmas time, of course. So I'd like to just introduce a little bit more about the experiments themselves first, and then I'll go on to say how the long-term treatments in these long-term experiments have created, as I call it, gradients. So in three chief areas, we've got gradients in soil fertility, chiefly through application of fertiliser or manures. We've got gradients in soil organic matter on the experiments, again through long-term application of organic amendments. And we have gradients in soil pH. And then I'll move on to talk about the kinds of science that's been done on the experiments that's looked at the effects of some of these gradients on various functions and services that I've called it, the things that we ask agricultural systems to do, chiefly net primary production or to support crop growth and crop yields, but also the role in particular of soils in regulating water and air, agriculture in terms of biodiversity support, and aspects of climate change, and agriculture as a means to either mitigate or which could potentially accelerate changes in the climate. And then we'll finish off with a little bit of a snapshot about how the long-term experiments are today and what the future might be. So if we go back to 1843, that's the given founding date of Rothamsted as an experimental station, even though Lawes had already begun some very small-scale trials. But 1843, with the arrival of Gilbert, they began to formalise their experimental trials in many ways. So they were very interested in the effects of nutrients on crop growth and how nutrients cycled through agricultural systems, through the crop and the soil. And they began to set up a number of experimental trials on the site here at Rothamsted. And they were probably intended to be demonstration trials that farmers could have a look at and demonstrate the effects of some of these new fertilisers on crop yield and how they cycled. Most of the experimental trials they set up, the treatments were actually very similar. They were looking at nitrogen, phosphorus and potassium chiefly. Separately, they had systematically kind of different plots separating out these three, but also in combination, and nitrogen in particular looking at different rates of application of nitrogen. So the treatments on these experiments were actually very similar. What differed between them was the test crop. Now, we already knew in the mid-19th century about the importance of rotating crops, and crop rotations were well in evidence in those times. There was a classic Norfolk four-course rotation, which had like winter cereals and legumes and root crops. But Lawes and Gilbert were looking at setting up experimental trials. So what they wanted to do was to carry out the same treatments on the same plots for a number of years to build up an evidence base, and with the same test crop. So they established a number of trials, each focused on a single crop. So perhaps the most well-known experiment on the site at Rothamsted, the broadbalt winter wheat experiment, begun in 1843. Lawes and Gilbert's experiment on a winter cereal. An experiment on a spring cereal, Hughesfield spring barley, begun in 1852. And I should say the names of these experiments follow the names of the fields, the convention in the UK for farmers to actually name their fields. And perhaps the other well-known experiment at Rothamsted, park grass, where the test crop here was grass for making hay. But the experimental treatments were actually very similar on all of these experiments. Well, in total, they established 9 trials on the site between 1843 and 1856. And some of these have been greyed out where they've finished or come to an end or been paused, but essentially 7 of them are still in the ground today. 2 of those 7 are not actively used so much, they're mothballed, but the other 5 are. So we have 5 of these classical experiments still in the ground at Rothamsted. And they're referred to as the classicals, the classical experiments. Some other experimental areas on the land at Rothamsted started during the time of Lawes and Gilbert as well. A couple of experimental areas which have slightly grandiose title of wilderness, which is always amusing when you meet visitors, particularly from the States and people like that, to refer to these as wildernesses, but they're land that were taken out of cultivation and now support natural regeneration woodland. And they were begun in the late 1880s. And a couple of other experimental sites as well. And 2 other facilities at the site also have their origin in the times of Lawes and Gilbert. That's the Rothamsted Sample Archive, which I shall mention a little bit later on, and the Met Station. So Lawes and Gilbert first started collecting rainwater in 1853. So we have one of the oldest weather stations in the UK at Rothamsted as well. And probably most of you know that you have a record breaker here, only about a mile away from where we are just now. So Broadbook is the world's longest running scientific experiment, and that's cited in the Guinness Book of Records. So since the time of Lawes and Gilbert, other long-term experiments have been established in the 20th century. At Harpenden, I'll refer to 2 complementary experiments, one called Highfield, one called Foster's. These are lay arable experiments, and they have treatments of long-term permanent grass, long-term permanent arable, also a long-term permanent bare fallow adjacent to Highfield, and also lay arable rotations as well. A long-term liming experiment begun in the 60s, finished in the 1990s but resurrected more recently. And then Rothamsted also has an experimental site at Woburn, very much near Woburn Park, Woburn Abbey, on a very different soil type, very sandy soil. And we have 2 continuing long-term experiments there, begun in the 1930s and the 1960s, and another long-term bare fallow patch of land as well. So now we'll talk a little bit about how these gradients have been established over time through the application of long-term treatments. So first of all, we have gradients in soil fertility arising from application or non-application of fertilisers and organic manures. So, for instance, I know the writing is very small, but what we have on the left-hand side, two graphs. We have time along the x-axis from the 1840s to 2020, and these are showing, the top one is showing phosphorus in the soil, and the bottom one is showing potassium in the soil. So through long-term applications of either phosphorus or potassium, we come to 2020 and we have these various range of soil phosphorus or soil potassium through repeated applications in broad bulk. Some experiments have kind of had the opposite. So a case here where an experiment up until about 1900 always had phosphorus applied annually every year, but then that application was stopped. So the crop every year has been mining, if you like, phosphorus from the soil that was already there, and as a result the soil phosphorus levels are declining over decades. And this experiment is literally referred to as the exhaustion land. It's looking at exhaustion of crop nutrients, particularly phosphorus and potassium in the soil. And then we have gradients in soil organic matter, chiefly from application of manures, but also from land use change. So in terms of manures, again we have a time series of points here where we've got soil organic carbon on the y-axis here, and we can show with the brown circles an increase in soil organic carbon due to repeated annual applications of farmyard manure every year. The green dots are that tiny part of the field at Broadwater which was taken out of cultivation from the 1880s, now supports grass and woodland, and that has very high organic carbon content as well, compared to the yellow circles at the bottom, which are the plots in the normal arable inorganically fertilised treatments. We have an experiment at Woburn, which is referred to as the Woburn Organic Manuring Experiment. This has had distinct phases of organic amendment. The current phase has been going for about 20 years now, and it has different plots where there's been applications of green waste compost or farmyard manure or chopped straw, or recently anaerobic digestate as well. So organic sources of minerals which have been applied. And again, this has shown soil organic carbon content, and we have a spread of soil organic carbon deriving from applications of those organic materials. And then also we can change soil organic carbon content by changing the land use. So we have these complementary lay arable experiments, as I mentioned, Highfield and Foster's, and these have long-term grass, long-term arable. But the key difference between these two is the starting point. Highfield was historically always under grass before the experiment was started. Foster's was historically under arable before the experiment was started and the plots were set up. So the control land uses for those two fields, you can see, are very different in their organic carbon content, much lower under the arable system compared to the permanent grass system over time. But they've had treatments where they've swapped those two land uses in each. So for instance, in Highfield, historically under grass, the plots that have been cultivated and now have arable winter wheat crops every year, and you can see a decline in organic carbon content. Contrast that with Foster's, where land has been taken out of cultivation, sown to grass in 1948, and we've got build-ups of soil organic carbon there. So we've got different trajectories of soil organic carbon in some of these experiments. And then the last gradient is that in soil pH, chiefly controlled by liming, and also the potential acidifying effects of fertilisers. And the experiment which most shows that, I guess, is the park grass experiment, which some of you may know because there's a public footpath that passes it. It's a mosaic of different grass species as they're responding to differences in soil fertility and soil pH, which I'll talk about a little bit later. But Lawes and Gilbert, when they started this experiment, which was looking at production of grass for making hay, they found that applying nitrogen did increase yields of grass, but it actually had an acidifying effect on the soil as well. So we have pH on the y-axis here, and we can see that some of the pHs were beginning to decrease when the nitrogen had been applied. And this has been in part corrected by deliberate application of lime. The plots have been split, and we've applied lime to half of the plots, but not to the other half, so we can correct for soil pH. But again, the same, we come to the 2020s and we have these very wide variations in soil pH, for instance. And we have an even bigger range on something called the acid strip. And this is a 200-metre length narrow area of land under winter wheat. And the soil pH changes from nearly 8 to less than 4 over 200 metres. And that was due to almost accidental differential amounts of lime being added, more lime being added to one part of the field but less to the bit further away. And it's created this nice transect, and colleagues here have looked in particular at the effects of this on soil microbiology. So we have these gradients in soil fertility, soil organic matter and soil pH that have derived from long-term treatments of fertiliser and applications of fertiliser, applications of organics. So now over the next few slides we'll have a look at some of the work that's been done on the long-term experiments that's looked at the effects of these on various, as I call it, functions and services. And if we start off with what I think would be the most important function that we ask of agricultural systems, that of supporting crop yields. So we can have a look at the long-term records of crop yields on the broadblock winter wheat experiment. And the broadblock experiment has had 3 very distinct phases in its history. So it was first started in 1843, as we mentioned, and on the top right, an old aerial photograph shows that it was these very narrow strips of land that run the full length of the field, and the different treatments were applied to these strips, so nitrogen, phosphorus, potassium at different levels, and a farmyard manure plot. And all of the long-term experiments would also have a plot where there was no application of fertiliser or manures, a nil plot or a control plot. So what we've got here, we're just going to show 3 treatments here, 3 extreme treatments, I guess. Again, we've got time along the x-axis and grain yield on the y-axis. So the blue line is the wheat grain yield from those the nil plot. So it's never had any manure and no fertiliser added. The crop does still grow incredibly, so it is obviously harvesting something from the soil, and there are other sources of nutrients as well, of course. But obviously Lawes and Gilbert demonstrated quite quickly that you could increase yields by either adding farmyard manure, which is the red dots, if you can make them out, or if you added sufficient inorganic fertiliser, chiefly nitrogen, which is the green dots there. And in fact, the two are very similar yields from purely farmyard manure as purely inorganic. The crop was continuous wheat, grown every single year. There's some display cabinets up at Rothamsted, and this is one from, I think, the 1880s. So these old varieties of cereals, very tall, very long-strawed varieties of cereal. But if you notice, by the time we get to 1925, the yields are beginning to dip. And one reason for that, believed, was increased prevalence of weeds and increased problems with weeds. That point in history, of course, weeding would all have been done by hand hoeing. And Rothamsted Farm, in common with every other farm in the UK, if you think about the timing, just after the First World War, shortages of labour on farms, and that's what they believe might have actually led to some of these decreasing yields. So that actually triggered the first modification of the experiment. And what they did was to split those long strips into 5 And in any one year, one of those sections would have been kept bare fallow, which is a simple technique for controlling weeds and pests. And that would rotate through. So the crop is still continuous wheat, but actually it would have 4 years of wheat, then a year of winter of bare fallow, and then go back to 4 years of wheat. And you can see it had the desired effect. The yields did recover, but still this big gap between the nil plot and the farmyard manure or the inorganic fertilised plots. And we can see a couple of other innovations coming in as well: liming to correct soil pH, and introduction eventually of chemical herbicides rather than hand hoeing to remove weeds. And then we come to the current phase of the experiment, which has been here since the late 1960s. And the big change here, following the Green Revolution and advances in crop breeding, the shift from the longer, long-strawed, older varieties of winter wheat to these new higher yielding, shorter varieties of wheat. That resulted in virtually a doubling of crop yields, as you can see, this sudden jump. And then the broken lines also indicate the other main change that was made in 1968, and that was the introduction of a crop rotation to half of the field. And there where our greatest yields are today, it's for that first winter wheat crop following a break. So obviously rotating crops is more applicable to farms in general. So we now have an experiment where half of the experiment has a crop rotation, more akin to modern agriculture, but half of the field is still in continuous wheat. So that keeps the link back to 1843. So that's another common feature of these long-term experiments, where the initial plots have sometimes been split to keep the original treatment going and then to modify the treatment on the other half. Because when we're thinking about crops, yields, food, as well as yields, we also need to think a little bit about the nutritional quality of the food as well. And colleagues have looked at micronutrients in wheat grain. What we have here on the top left, it's essentially this same yield graph again. It's the solid blue line and the solid green and red line from the 1840s through to the 2000s. We've just got it expressed in kind of black and white symbols here. So the black symbols at the bottom are the nil plot, and the other two are either the farmyard manure or the inorganic fertilised plots. And again, it's showing that sudden increase in yield in the late 1960s that came from the variety change. But what about the concentrations of certain micronutrients in the wheat grain? Well, colleagues found that there was some evidence that they'd actually decreased. So they focused in particular on zinc, copper and magnesium, very important micronutrients for our bodies. And they found that as a result of increase in yields, we may possibly have diluted the nutritional quality. So that was an interesting aspect. Now, one potential reason for that could have been if there was insufficient in the soil itself for the plant to uptake. But they measured it in the soil and they found that there was actually no evidence for that. And in fact, it looked, if anything, like the concentrations of these three micronutrients in the soil was actually increasing rather than decreasing. So that's an interesting finding that colleagues found. Now, talk a little bit about the role of agriculture and in particular soils in regulating water and air. And one property of soils is their structure. So this is the arrangements of sand, silts and clays into aggregated units or clods or aggregates, you might call them, surrounded by the pore network. And it's the pores that are allowing water and air to move through the soil. So allowing water to drain through to groundwater, or allowing air to get in there as well. These are very old thin section images under the microscope of the same soil type from one experiment, but under different long-term treatments. The one on the left is soil structure under long-term permanent grassland. The one on the right is soil structure under long-term permanent bare fallow. And you can see that they're very different. Much greater porosity in the soil under grassland compared to that under bare fallow. And they have vastly different soil organic carbon contents. And that has an important role in terms of how soils are stable against certain stresses. So this is going to be, if it works, and it should work in a moment, I'll just show a very short video clip here. What we're looking at is 2 Petri dishes full of water just from above. What we're going to do in a minute is drop an air-dried aggregate into each of those, but one aggregate's derived from under long-term grassland and one is derived from under long-term permanent arable with vastly different soil organic carbon contents. And we can see what their behaviour is when we drop them into water. So this is filmed in real time, there's no tricks here, but it just illustrates, you can see that that aggregate from under grassland is stable to that water stress, whereas the aggregate taken from the permanent arable or long-term arable was completely unstable and slate. So the way soils are managed has an impact on their soil organic carbon content, and that has an impact on certain soil properties as well, like strength and stability. Colleagues have looked at this in a bit more detail. They've taken intact soil samples from various treatments on the Broad Balk experiment, and they've taken very high resolution images of them using X-ray CT techniques. So a bit similar to in the hospital, the X-ray CT can take an image of a part of the body in very high resolution. So you can image soils as well and see at very fine detail their structure and their pore structure. And colleagues have found that there was a nice relationship between the amount of pore space and crucially the amount of pore space that's connected to each other, and on the y-axis the ability of soils to transmit water, or its hydraulic conductivity, to give it its scientific name. And they found this nice relationship, and they could also relate it to these long-term treatments as well. The diameter of the circles there is in proportion to the soil organic carbon content of those samples. So they found the soils under the long-term woodland or grassland, or the farmyard manure treated plots had a much greater soil organic carbon content, had much greater connected porosity, had much greater ability to transmit water through. And they found similar with those extreme treatments at the Highfield lay arable experiment as well, in terms of grass, arable and fallow. Moving on to biodiversity support. The parkgrass experiment is a really nice one to look at effects of soil fertility and soil pH on botanical diversity. So the parkgrass experiment, established in 1856, very close to Rothamsted Manor. It was established in a species-rich meadow, so there would have already been quite a lot of diversity of grass species in that meadow. And Lawes and Gilbert set up their plot-based experiment in 1856 and started to apply their different fertiliser treatments. And they found very rapidly that changing the fertility of the soil through these applications of these fertilisers had an amazing effect and very quick effect on the seed bank, how the seed bank responded to that. And how the seed bank responded is kind of illustrated in this schematic here. Where you add fertiliser, particularly nitrogen, they found that yes, it did increase the biomass, the yield of the grass crop, but it tended to favour a smaller range of very competitive grass species that could maximise from that nitrogen and outcompete a lot of other things. Compared to on the left where you don't add nitrogen, so you have a less fertile soil, you get much lower overall biomass or yield, but you get a much greater diversity of botanical species there. So as you can probably see from the aerial, and if you have wandered past, particularly in kind of May, in the springtime, you'll see it's this mosaic patchwork of different grass species, which you can pick out when the grasses are in flower. On top of the soil fertility on park grass is the soil pH. So there's fertility and pH, and the combination of the two has this amazing effect on botanical diversity. But just to say, plant species richness greater in the less fertilised plots, less in the more fertilised plots. But the pH also has an effect as well on both yield and the diversity. Coming a little bit smaller in scale, colleagues have also looked at effects of long-term treatments on soil fauna, microbial biomass. The left-hand graph is mainly looking at some invertebrate numbers under long-term grass, arable and fallow, and they found that generally the invertebrate biomass increased in order of fallow arable grass, so it's kind of following the soil organic carbon content of those soils and the amount of disturbance those soils are subjected to. On the right, some work that colleagues did on looking at earthworm numbers, which is never going to be great on the broad bulk experiment because it is an annually cultivated experiment, which is not so great for earthworms, of course. But nevertheless, they could see much greater earthworm populations under the treatment that receives farmyard manure, for instance, and also one of the sections that has the straw incorporated into the soil as well. Okay. These are just some graphs showing the effects of soil pH on microbial biomass as well. The left-hand panel of graphs are taken from the long-term liming experiments, both at Rothamsted and a complementary one at Woburn. And again, they're showing this nice relationship of increases in soil microbial biomass as the soil pH tends from being quite acidic to close to neutrality. The top graph shows the basal respiration, so that's the activity of the microbes as well. And again, that's showing an increase. And then the middle panel is showing a very similar pattern in this acid strip, which is under winter wheat as well. So it's shown an increase in bacteria and fungi as the pH increases from acidity to neutrality. And colleagues have also looked at the diversity both of bacteria and fungi at different soil pHs in the park grass experiment, comparing soil that's maintained at pH 5 and soil that's maintained at pH 7. Very little differences actually in the bacterial communities, but the fungal communities, hopefully you can see that there's a greater diversity. There's more colours in those bars on the right hand side for the fungal communities in soil that's maintained at pH 7 compared to that that's maintained at pH 5. Climate change. So one thing Rothamsted can contribute to, I guess, is evidence for changes in climate. So this is Rothamsted's weather station, 5-year mean air temperatures at Rothamsted. Air temperatures first began to be recorded in the 1870s. So there's obviously a lot of year-by-year variation, but we certainly seem to have quite strong evidence, certainly since the late 1980s, 1994 increases in air temperature at Rothamsted. And you may see these climate stripes, which is quite a nice way of showing these kinds of trends in climate. It's quite a neat way of expressing that. So agriculture has a role to play. It can potentially accelerate or cause problems, but it can also perhaps provide a few solutions. The one thing agricultural systems might be able to do is to sequester carbon. So the idea of this is to take carbon dioxide out of the atmosphere through using plants and through the root system to actually deposit it into the soil where it might be more stable. So again, because of these long-term experiments and long-term treatments, we contribute to this debate and see what the evidence base is. On the left-hand side here, again, we've got time series, and this is looking at annual applications of farmyard manure in 2 experiments, Broadbrook and Hoosfield spring barley. Annual applications of farmyard manure, and we can see that soil organic carbon contents have increased. But I think in both of these graphs it's important to note that it does look like they're plateauing. And these are arable soils, they're annually cultivated, they have an annual crop. They don't have a perennial crop and, you know, no-till or anything like that. So it is demonstrating that they probably will hit an equilibrium whereby inputs of carbon increase equal outputs as well. So it's kind of saying that the sequestration of carbon in soils may not carry on indefinitely. Each soil will have its own capacity to store carbon. Another potential solution, of course, is to take land out of cultivation. Of course, you lose land which could produce food, but these 2 long-term woodlands at Rothamsted that were taken out of cultivation in the 1880s, we can see build-ups of carbon in the soil beginning from the 1880s and then through to 1999 was the last point in time here. Mainly in the topsoil. These are depths. We have depth on the y-axis and carbon on the x-axis, but also some carbon being stored at lower depths as well. And Rothamsted colleagues also a few years ago looked at some of the different treatments on the long-term experiments to see how much carbon they're sequestering on a per year basis, and compared this to a kind of target which was part of one of the climate change conferences, and the one in Paris in 2015 did some calculations whereby they estimated how much carbon soils would have to sequester to offset the increased carbon emissions from all of the sectors. So we can look at some of these long-term treatments in our experiments and see where they lie, whether they're sequestering carbon above this target or below this target. Not to say anything about the feasibility or sustainability of adopting those at the larger scale, mind you. And also linked to climate change as well, greenhouse gases, a lot of focus on carbon dioxide, but there are other key greenhouse gases as well. And one notable one is nitrous oxide, because I think agriculture is the biggest source of nitrous oxide emissions. And nitrous oxide as a greenhouse gas is approximately 300 times more potent than carbon dioxide. So again, colleagues have collected gas samples emitted from various treatments on Broadbrook and have shown that as you increase nitrogen applications, you do run potential risks of losing some of that nitrogen as nitrous oxide, and also from application of farmyard manure as well. And then final slide with some data on it, and then we'll begin to wrap things up. The long-term experiments have sometimes been sometimes been used to look at other aspects as well. So there was a lot of work done in the 1980s and 1990s on nitrate. There was a lot of interest in pollution of fresh waters, eutrophication, runoffs of nitrates and phosphates getting into rivers and causing problems. So we have a facility at the Broadbrook experiment where we can collect drainage water from these differently treated plots. And sure enough, as you may imagine, some of the plots that have very high inputs of nitrogen as fertiliser, we can actually measure that there's a reasonable amount of nitrate that's coming out of drainage water. So fertilisers are obviously important, but if you fertilise more than the crop can uptake, then you run the risk of losing it either in gaseous forms or potentially to drainage waters as well. And then quite a recent study was looking at microplastics. So a student at Lancaster was interested in microplastics in agricultural soils. So got in contact with Rothamsted because he knew through our sample archive that we got a time series of soil samples collected from before the plastic era and then since plastics have been more widely used. And he was able to demonstrate that there have been accumulations of microplastics, particularly in soils that have been long-term applied with mineral nitrogen and also farmyard manure as well. He did actually find increases in the nil plot as well, so there are obviously other sources of microplastics in agricultural systems as well. So, all of which brings us to the present and then perhaps into the future. So, you may have noticed this little logo in the bottom right of all the slides. The Biotechnology and Biological Sciences Research Council. So that's the part of government funding that provides strategic funding to Rothamsted and other such institutes in the UK. So their remit is to support biosciences in the UK. So they do support some of Rothamsted's big science programs, but they also support the long-term experiments as well. And they support us, they fund us as something called a National Bioscience Research Infrastructure. It's a bit of a mouthful, but the clue is in the title that they're funding these long-term experiments because they see them as national resources. They're not here just to support Rothamsted science, but they're here predominantly to support UK bioscience and increasingly international science as well. So they support us, so that allows us to maintain the long-term experiments. They also support us to maintain our sample archive, which we're adding to every year because, as we mentioned, some of these experiments are still in the ground and we're still sampling them and analysing them. They support something called the Electronic Rothamsted Archive, which is essentially the database, all of our data, which we're increasingly making completely open access, and it also covers us to continue our environmental monitoring work, chiefly maintaining our weather stations, but we also are part of various UK networks as well that are monitoring the environment. And we're seen really as a research platform in current speak to support new research in three main areas: sustainable agriculture, food security and climate change. They're the kind of hot topics that we think we can support new research of. And a lot of our activities are kind of illustrated in this montage on the right hand side, whether it's collecting yield data, archiving samples, producing data sets, publishing and public engagement like tonight. We have to report regularly to the funders. So here's some data from our latest reporting year, which runs like April to March. So we're collecting a lot of yield data from these experiments. We're collecting a lot of samples, a lot of individual analytical data being generated, literally runs to tens of thousands every year. And we're adding to the sample archive. And then the middle set really says about how we're collaborating. We often get approaches from people who want to work with us, maybe want to measure something on the experiments or to sample from them, or to interrogate data sets that we already have. And as I mentioned, we do quite a lot of public engagement. We host quite a lot of visits and increasingly a lot of international visits as well. So we saw nationals from at least 19 countries in that last reporting year. And we're not standing still. So in the last 7 or 8 years, we've established 2 new experiments which are intended to run for a long time. One here at Rothamsted, one at our experimental farm in Brooms Barn, which is in Suffolk, between Newmarket and Bury St Edmunds. And these are looking at different crop rotations, different tillage, different application or non-application of organic amendments, and various smart ways to protect crops rather than just chemical means as well. So there are new experiments that have been set up, which hopefully in 180 years, who knows, they might still be around. So just to summarise about what the main things that we've learnt. I think we've learnt that we can sustain crop yields, soil fertility through use of fertilisers and organic manures and liming as well. But obviously injudicious use of some of these inputs can have detrimental effects. I think everyone would probably know that. We report on this quite complex interaction between nutrition in the soil and nutrition in the food that we eat. Soil carbon, very key. It's greatly affected by the way that soils are managed, and soil carbon really has this key role in controlling a lot of the things that we want soils to do. But I think we can demonstrate that the long-term experiments, they continue to support new science. And as we say, these three hot topics: sustainable agriculture, food security and climate change. So I think we can be confident in saying that they are as important today as they ever have been, really, and they are hugely valued. So we're really indebted to everyone from Lawes and Gilbert onwards who've managed to maintain these experiments and pass them on to the next generation, and eventually I will do at some point, I think. Thanks to my current colleagues and in particular everyone who's worked from us from outside of Rothamsted. And of course, thanks to the funders for supporting the ongoing work. And thank you very much for listening.
Thank you, Andy, for the great talk. Do we have any questions?
I'm interested in the bit that says environmental monitoring. So, do you do that all over the site?
So I guess the main thing I'm thinking there is we have our weather station, so we have just one here, but we also have ones on 3 other sites as well. So Woburn Bridge.
Around the site, digging balls of ground and see what's happening.
Not as such. So one of the main things we're involved in is the Environmental Change Network. Which is basically the UK's environmental change network. There's a load of freshwater sites that are included in that, but we're one of the terrestrial sites. So that includes either weekly or fortnightly or monthly monitoring of various environmental chemistry parameters. So for instance, the River Firth flows through the site, so we collect river water every week and test for the chemistry of that. We also have various diffusion tubes around the site. So there is a little bit of environmental monitoring, yes, that goes on around the site.
Okay.
Yeah.
Yeah, I was just because I've seen the funnels and the light boxes with the motor with everything on.
Yes.
I was just wondering, because when you are standing, for example, if you have your back to the manor house and look out over the fields toward the Whitehorse, until a few years ago there were sheep on there.
Yes. Yeah.
And now there's dog poo, essentially. So I was just wondering if you, by any chance, were monitoring that and seeing any differences, the effect of, the greatly increased footfall compared to how it was 10 years ago.
No, I don't think we have, but I think you're right that the footfall has increased, and certainly in the pandemic and since. The sheep, I think we used to rent out the fields basically to local farmers for monitoring the sheep. And you mentioned something else a minute ago. Oh yeah, the light boxes and the suction traps. So that's part of the Rothamsted Insect Survey. So again, that's another of these National Bioscience Research Infrastructure that BBSRC fund. And Rothamsted is kind of the home network of the country's insect survey. So they are also carrying out various monitoring of insect populations. The light boxes for moths, the suction traps mainly for aphids, aphids being an important pest for crops.
Are you going to do the experiments, although you're tracking again of the bees? Do you remember when there was a big radar, like a huge big satellite dish that was being drove back and forth every day, and you were tracking bees with it, which is absolutely fascinating.
Yeah, that scientist has moved on, unfortunately. But I think we looking at Georgia here partly. I think we have got some equipment from a university, a project with Leeds, I think, who have had a similar setup. Yeah.
That was amazing to look at.
Yeah.
I've got a couple of questions about the wildernesses. Do you include the wildernesses in the category of long-term experiments? Are you happy to talk about that?
Yes. Yeah. I mean, I would. Yeah.
Questions like when you mentioned them on here, you said the last measurements were 1999. Is there any plan to see what's happened in the last 25 years? Yes,
yeah, there is. And in fact, I haven't got round to it yet, but we did soil sample there in 2021, I think, 2021 or 2022. So there should be another line to draw there on the Broadbort wilderness. And the Geescroft is on the to-do list to do again. So yeah, we do need to do that because it has been literally 25 years since we've updated that.
And any plans to measure the above ground carbon?
I think we should do, because it's been done before, and it would be useful to know because it's a bit small, but these coloured graphs in the middle, the brown is the organic carbon in the soil, but the other two are the above ground, either the litter or the biomass, which is obviously an estimate. But I would like to do it. Yeah, I think I'll probably have to consult my predecessor to see how they went about it.
The papers I've read about how they went about it involve equations which subsequently have been criticised and against measuring by laser to try and get the volume of the trees. So I wonder whether remeasuring it with more accurate methods now will considerably change the old measurements.
I think, I mean, I think we should use whatever's the best technique today to do it, and we can attempt to kind of calibrate backwards. But yeah,
because the paper I read said they were out. The old equations were out by 50-60%.
Yeah.
So really,
just
a long way.
Yeah.
Potentially.
Yeah, something to look into.
This was great, thank you. It's interesting, isn't it? Because like farming, food, and kind of climate change. If you wanted to pick three incredibly controversial hot topics, you're there in all of them really. And so I'm just wondering, and also ones where, let's say, there's not necessarily a lot of rationality in the debate around those things. And I'm just wondering, therefore, what's the mechanism by which, the hard evidence and the scientific insights that you generate here get translated into policy, get translated into agricultural practice?
Yeah, I think we do have some inputs. I mean, I've not done it myself, but colleagues have certainly been before kind of select committee or committee type stuff in Parliament presenting evidence. We also had a couple of years ago the Environment, Food and Rural Affairs Committee visit us here looking for evidence. I think that was on soil health, for instance. I think there is sometimes a challenge between the scientific process and publication, and perhaps the translation of that into policy is not always easy, I think. But I think we do have links, and we do have the mechanisms, I think, to try and make this a bit more public. And yeah.
Good.
Yeah.
It's about the microplastics that we get in soil. Is that absorbed by plants and we eat
That study was only looking at the soils, and they didn't look at the crop samples as well. So it remains a bit of an unknown, really.
How does it get in there in the first place?
We're not entirely sure. I mean, we did have these increases in the nil plot as well. So I don't think it's it can't be as straightforward as only being in the materials in the fertiliser itself. As far as I know, we don't have the kind of coated fertiliser. I don't think that's ever been applied on our experiments, but nevertheless it is there. But there could be sources from, you know, from the tractor tyres, from plastic packaging that fertilisers are contained in, that probably some aerial deposition as well, although it's the word micro, in actual fact, in relative terms, it was quite large. So I thinking myself, I wouldn't have expected that to have been aerially deposited, the kind of size classes that he was looking at. It could have been perhaps, but there's all the other kinds of inputs of agrochemicals, pesticides, herbicides, so there's probably a few sources where it could have come from. Yeah.
Hi, just wondered, have you conducted any bioarchaeological experiments ever on the site to look at kind of what might have been growing 1,000 years ago, 2,000 years ago?
No, I don't think we ever have. No. Could be interesting. As far as we know, it has been a long-term arable site going back many hundreds of years, as far as we can tell. But no, we're thinking like pollen analysis and that kind of.
I'm an archaeologist and I've done a bit of work by the mausoleum, and it was a Roman rural estate definitely, but probably also an Iron Age estate before that, and then probably for a very long time before that as well.
Mm-hmm.
So I was just wondering whether anyone had taken any soil samples at that level.
Not to my knowledge, no.
Yeah,
If you'd be interested.
I'd be so interested.
Okay.
Just because actually it ties into what you were saying about climate and what's growing there and why it's growing there. I have got some ideas about what might have been there, because there has been some bioarchaeology down in sort of around Verulamium. So it was barley, emmet, I think that's the right word. It was barley and a very early other kind of wheat. And then also I think there was quite a nice division between pasture land and land taken over for arable. It'd be interesting to see whether it carried on and it was similar up to the estate up here as well.
Yeah. Yes.
Not for any particular-
Sounds like a project proposal in the offing.
So-
Yeah.
Any other questions?
Some of your data ranges showed you moving from acidic to neutral. Is there an environment beyond neutral in the alkali spectrum? Does that just not exist in nature?
You can get alkaline soils, yeah. I think most soils tend to be slightly acidic, but the optimum for crop growth probably is around neutrality, 6.57. When it gets much below 6 and 5.5, crops do suffer. But if you go too alkaline, you can run into problems of nutrients getting immobilized, particularly phosphorus, that the plant can't actually extract it because it's kind of held so tightly chemically. The soils are generally fairly neutral here, in the sense that we are on chalk a few meters below us and clay's on top of that. But I guess the main acidifying effect can sometimes be long-term applications of nitrogen fertilizers, particularly ammonium sulfate, which can cause real decreases in pH.
Experiments are neutralising the effects of those fertilisers rather than seek to push the soil into alkaline and see what happens on the alkali side.
Yes, I guess there hasn't been any experiments where we've pushed it much over neutrality, I guess. Yeah.
So you said it's essentially growing a wheat crop year after year. Do you change the cultivar for this wheat crop? And if so, how do you decide which cultivar to put in? I'm guessing that's a great debate.
It is, yeah, by committee basically. If I can bring it up.
I'm guessing it's sort of like you're looking at the weather soil is at different times of the drilling. You know, if you think you're going to have a very weedy year somehow, or a pesty year, and you want certain resistant crops.
Yeah, yes. So it's in tiny writing, but at the bottom, it's not all of them, but these are the main varieties that have been grown on the experiment. In small writing, the names of them. Tiny writing, yeah.
They're different each time?
No, so what we would do now is, because the experiment is, half of it still grows continuous wheat year on year, and the other half is in a 5-year crop rotation. So what we would be looking to do is to run with the same variety for at least 5 years, so we get kind of a full cycle, because it kind of rotates in different parts of the field, if you like. So when it comes to changing the variety, we always look for one off like the recommended lists from AHDB, and they're all bread milling wheats, I should say. They're not feed wheats, so they're all high quality bread wheats. That's what we've tended to do. There's only been one since the 60s. I think there's only been one that's been a non-bread making wheat, but all the others have been. So we're looking for something that looks to have good properties like resistance to diseases. Another consideration is, will it be around for 5 years? Can we get hold of seed? We wouldn't want something where we could get it in the first year but then not be able to get it otherwise. So they're the kinds of factors that come into it. And so take advice from the farm as well. So this is going to be the last year of this particular variety, which is Zayat, which has been on, I think, for about 6 years now. So it is ready for a change. But we met last year to discuss it, and the consensus was there wasn't one that we were like fully behind. We thought we'd give it another year of tests at farms all around the country and see what the recommended list is next year, and then we'll make a change.
So are you using farmers around the country that have got similar soil conditions to our one here, or climate, to help determine which cultivar to pick?
I think there's a bit of that, yeah. I mean, certainly the farm staff can certainly advise us a lot on that kind of thing, but mainly it is looking at the recommended lists, and they're based on trials from around the country.
Okay.
Yeah. And just while I remember, kind of links in with climate change, I guess, a little bit. It's a winter wheat experiment, but twice in the last 10 or 15 years we've had to sow a spring crop on broadbort because it's been too wet in the autumn to be able to sow a winter crop. So I think there are evidence for different weather patterns as well, perhaps wetter autumns, wetter winters as well, and that's actually impacted this.
What do you rotate it with?
So on the top right, you might be able to see some symbols on the rotation sequences. So the crop rotation has occasionally changed as well, been modified, which I think is good. I think one thing about long-term experiments is we don't want to kind of set them in stone. We want them to be relevant, but kind of protect their long-term integrity. It's a bit of a balancing act. Current rotation came in about 2017, so we have 2 years of winter wheat, then a year of winter oats, then another year of winter wheat, and then winter beans, and then round to the start. And so it's a 5-year rotation, and there are 5 sections on the field that are in that rotation. So it means when you go up there, in any one year, there will be every year of the rotation represented. So you will see, in this schematic, you could probably see the light green stripe is winter oats, and the slightly different shade green, about 3 up, is winter beans. And if we were to take that aerial photograph the next year, the beans and the oats would be elsewhere. But before that, the rotation before that, we had forage maize, for instance. We've had potatoes in there in the past. We've had years on bare fallow, so it would go wheat, fallow, potatoes. But keeping land bare fallow is not seen as a good thing to do, to leave soil bare over the winter and not to have a crop on there at all.
So I think you have a soil sample bank, is that correct? So, and that's been over a long period of time. How do you keep those samples so they stay stable over a long period of time? Do you freeze them, and if so, does that change the properties? And you analyse over time, and does that shift or vary over time?
Yeah, so the samples in the archive are all dried in some way. So the soil samples have been air dried and crumbled. And that's good for most of the things that Rothamsted's been interested in, which has been mainly focused on nutrients and bulk chemistry. It's not always so good for looking at other aspects like microbiology. You probably don't want to dry your soils. Or if you're interested in structure at a larger scale, you don't want to have ground them and put them through a sieve. So it does have its limitations, but for most of the applications, it's been seen as good enough. And people have found, for instance, that the DNA is still viable in the samples. So the soil samples are air dried and crumbled. Crop samples have been oven dried to 80 degrees and archived. Just seeing if I've got a. Yeah, some of you may have visited, I'm not sure. So it's as old as the experiments themselves. And there's over 300,000 samples in there. So it's dried. I think there's probably no ideal way of doing these things. If Lawes and Gilbert had had a minus 80 degree freezer in 1843, that would have been good, I guess. And colleagues who work on microbiology often take their own samples and prepare them separately compared to the kind of bulk archiving exercise.
Yes, a few years ago, a few years in a row, there were large bands of sunflowers that were grown in fields, and I think 2 years ago there was a cornflower field or something. So, why?
I think that was I think it was a kind of scheme, wasn't it, that the farm. Feel free if you want.
I made a face, didn't I? They were different, and they weren't big within the rotations. So there were different ones. They were closer to the tennis courts, and over by the. Oh my gosh.
So towards the farm as well. I think it's back over there.
Yep.
Yeah.
So there was linseed. There's been a few rotations, but those are mostly on the actual farm plots rather than the long-term experiments. So there was a trial to try and boost birds. And
Yes,
so
it
it's-
worked in my garden all over the place.
Yeah, there was those, but those are sort of separate from the rotations that were
Yeah.
happening there. Yeah, so there was sunflowers in 2022, I think.
Yeah.
And linseed down towards the oh gosh, the White Horse Pub.
Yeah,
Whatever that.
the green field that was there.
Yep.
Yeah.
2 years ago.
That was linseed.
It was gorgeous.
Yeah.
I think they've had some margins that were kind of part of agri-environmental schemes as well, I think.
There was, cornflower was a field strip, and that was to try and determine whether or not pests and beneficials could be monitored and brought into the fields. So there were strips through the center of the field to try and figure out whether or not you could bring pests and beneficials in, and to try and monitor where they were going. If you had the strip up the middle of the field versus around the edges of the field. Sorry.
Thank you. No. My weed ecology expert colleague.
Gloria,
Yes, I was thinking back to when these long-term experiments were initially started, and I don't know if you know so much about the public perception of them when they were first maybe proposed. Did you think, because obviously someone's going to say like, did they start off with the idea of, oh, we're going to have this experiment with these treatments and we're going to keep it going for as long as possible? And how were they going to fund it? Or do you think it started off as a, it's been like this for a while, we can still keep it going like this, and then the questions and the hypotheses came later?
Yeah.
I'm trying to sort of work out how did it start? What was the big idea in the beginning?
I think the big idea, one of the big ideas was a debate about nitrogen and how crops acquired nitrogen. And there was two schools of thought, and probably the most famous agricultural scientist on the continent was Liebig in Germany. And he changed his mind, and he eventually came to think that, 80% of the air is nitrogen. That must be how crops acquire nitrogen. And there'll be no benefit to adding nitrogen to the soil because there's enough in the air. And Lawes came to think that no, if you add nitrogen to the soil, the crops will uptake it through the roots and you'll see effects on crop yields. So there was quite this debate between Lawes and Gilbert and Liebig in Germany, and a lot of very acrimonious letters between the two, all played out in public, all published letters. So I think there was a few things. I think Lawes was actually, and Gilbert, they were very meticulous in thinking we should run with these for a number of years and build up a good evidence base because of the seasonality and that. I think there must have been a stubbornness to prove that he was right against Liebig. I think that was another issue as well. And I think as kind of visual demonstrations as well, I think there's evidence that Lawes hosted a number of farmer groups and that who would come to Rothamsted to see these experiments for themselves and to talk to Lawes and Gilbert.
Mind the fact that he had a factory that produced artificial fertilisers people.
He had a stake in the game. He did have a stake in the game. I'm just trying to, but he had this quotation, which I'm wondering if I've got or not. He had this quotation, I think, when they opened the first laboratory, which said something like, the aim of these experiments is not necessarily to put money into my pocket, but to give you the knowledge to be able to put money into yours. So yes, he did have a stake in the game to demonstrate, in particular, superphosphate, the effect it would have. But I think he was genuinely open about the findings that he had and to demonstrate the effects of these things. He did self-fund it all the way through to the early 20th century. I think it wasn't until the 1910s or 1920s that there was any government money put into the research at Rothamsted. It was all funded by Lawes and Gilbert, and apparently he set up the Lawes Agricultural Trust, which continues to this day. It's the charitable body that owns the site. I think the story goes that he didn't trust his son with the inheritance, so he set up the trust to keep the institute going.
Can we have one more question, please?
There's a lot of innovation around biofertilizers and how they can increase the amount of chemical fertilizers supplied on the soil and to the crops. And I was wondering if it's something which is on your radar at all to try on experiments that you have at Rothamsted.
It's probably not currently on our radar, but it could be. And I think, as we mentioned, I don't think these should be set in stone, and they should evolve if there's good scientific reason. And we do have some flexibility with some of the experiments that we could introduce new methods, new inputs as well. So it is something we could consider, I guess. Another thing sometimes put to us is there's a lot of technology going into gene-edited crops, for instance. Could we grow some of them? So yeah, I think we should always try and keep these things relevant, and it's that balance between keeping them relevant and kind of protecting their long-term integrity. But there is some scope to do things like of that kind of nature, I think, yeah.
soil as well.
Yeah.
So I think Rothamsted are looking at it just not from a long-term experiment treatment point of view.
I think those new rotation experiments, they have cover cropping, and we've got cover cropping as one of the treatments at the Woburn organic manuring experiment as well. So there's some work there, yeah.
Okay, Michaela.
So you were saying that you increase the yield, but the micronutrients are going down, even if they're present in the soil. Why do you think that is?
I mean, whether it's just a dilution effect and there's more carbon, more carbohydrate or something going into the grain rather than the nutrients themselves, I'm not so sure. But it's work that Steve McGraw's been leading in, so I can't quite remember if they'd got a theory as to the mechanism as to why that's occurred. Yeah.
So, in the interest of time, we will finish here. Thank you very much for coming, and thanks once again to Andy for giving a very exciting talk.